Assalamualaikum and hi everyone. So today we will present about biofilm formation in water piping system and its microorganism. My name is Nuriza Fikra binti Japri. My metric number is E24A3300. Now let's proceed with our first presenter.
Assalamualaikum and salam sejahtera. My name is Nurul Nasri binti Ismail and my metric numbers is E24A3482. And I want to talk about identify the microorganisms that form biofilms in water piping systems. Okay, biofilms form on stones in a stream bed, for example, but also on the inside of water pipes. A fact that can play major role in the hygiene of tap water. In simple terms, a biofilm consists of microorganisms such as bacteria, fungi, protozoa, or algae that are embedded in a layer of slime and deposited on a surface. These communities of microorganisms are very well adapted to their environment. This is because of the microorganisms organized in biofilms use metabolic properties of the other species. They cohabit with their protective mechanisms to multiply in biofilms. Biofilms in water piping systems are complex communities of microorganisms embedded in a self-produced matrix of extracellular polymeric substances, which is EPS. Microorganisms that form biofilms in water piping include bacteria, fungi, protozoa, algae, and higher organisms such as nematodes, larvae, and crustaceans. Biofilms can cause water quality and operational issues. They can also trap viruses and parasites, such as Cryptosporidium, which can attach to the biofilm after a contamination. I think that's all from my topics. Thank you.
Hello, my name is Nuratika Metatman. My metric number is E24A3406, and I would like to talk about microorganisms involved in biofilm formation in water piping systems and their unique characteristics. So let's start with bacteria, the most dominant players in biofilm formation. These microorganisms stick to pipe surfaces and rapidly reproduce, and they produce a sticky substance called extracellular polymeric substance, or EPS in short, which acts like glue. This protects them from water flow and disinfectants. So common examples include Pseudomonas aeruginosa, Escherichia coli, or in short is E. coli, and Legionella pneumophilia, which can pose health risks. We have fungi such as Candida albicans. So as we know, fungi are tougher than bacteria because they can withstand harsh conditions like temperature changes and disinfectants like chlorine. Fungi, they form strong, persistent biofilms that are much harder to eliminate, so making them a significant challenge in water systems. And finally, let's talk about protozoa like Acanthamoeba. While protozoa don't directly form biofilms, but they play a supporting role. They act as hosts for bacteria, like providing nutrients and protein and nutrients and protection, which makes the biofilm even stronger and more resistant to breakdown. Moving on, I'm going to talk about the habitat of microorganisms involved in biofilm formation within water piping systems. So let's dive right in. First, environment. Microorganisms need three things to grow, which is moisture, nutrients, and a surface to attach to. Slow or stagnant water flow provides the ideal conditions for biofilm formation because it gives microorganisms time to settle and multiply without disturbance. And next, we have pipe surface. Inside the pipes, microorganisms stick to the inner surfaces, whether the pipes are metal or plastic. They prefer areas with rough textures, cracks, or corrosion, which makes it easier for them to attach and grow. Over time, these surfaces develop a slimy biofilm layer and becomes harder to remove. After that, prime locations. Certain spots in piping system are particularly vulnerable. Dead ends or low flow zones in the network allow microorganisms to multiply undisturbed. Areas with low or inconsistent disinfectant levels, such as older or poorly maintained systems, are hotspots for biofilm formation. Finally, temperature plays an important role. Many microorganisms prefer warm environments, making hot water system or heat-exposed pipes more prone to biofilm growth. In summary, biofilms thrive in moist, low-flow, poorly treated, or warm areas within water piping system.
Biochemical pathway of biofilm formation. Introduction to biofilm. Bacteria are usually thought of multicellular. In nature, they frequently form surface-attached communities called biofilm. These biofilm are found on teeth, for example, dentin plaque, and on rocks in hot springs, among other places. It is because they cause equipment damage, product contamination, and medical illness. Biofilm are important in microbial pathogenesis and environmental quality. Pseudomonas aeruginosa in the lungs of patients with cystic fibrosis is an example of a single species biofilm. However, biofilm are sometimes made up of several species. Next, let's look at how biofilm formation.
Assalamualaikum warahmatullahi wabarakatuh. My name is Anur and I'm going to continue to present the previous slide. Biofilm formation. When bacteria sense environmental cues and adhere to surface, biofilm development begins. Appendages copulae have been used to adhere and form a sticky coating that draws in more germs. After being attached, they cease moving and shift around the surface using a technique known as twitching. The bacteria then create microcolonies as they grow and coordinate their action by exchanging chemical signals. For the result, the extracellular matrix, a slimy protective layer, is produced and to protect the germs from the damage. So then the biofilm will develop intricate structure with nutrient following route as it grows, so that bacteria can disperse and relocate its nutrients become scarce. One example for the biofilm is Pseudomonas aeruginosa, which is challenging to cure and can result in major health problems.
For example, the gram-negative bacterium Pseudomonas aeruginosa can form a single-species biofilm in the lungs of cystic fibrosis patients. Single-species biofilms are also readily formed in the laboratory. The first stage of biofilm formation occurs when a specific environmental signal induces a genetic program in free-living planktonic cells. In response to the signal, the planktonic cells begin to attach to nearby surfaces by means of flagella, pili, lipopolysaccharides, or other cell surface appendages. The attached cells begin to coat that surface with an organic monolayer of polysaccharides or glycoproteins, to which more planktonic cells can attach. As the cells enter into biofilm formation, they no longer maintain their flagella, and instead may move along surfaces using a twitching motility that involves the extension and retraction of a specific type of pilus. Ultimately, they stop moving and firmly attach to the surface. As more and more cells bind to the surface and divide, they form microcolonies and can begin to communicate with each other by sending and receiving chemical signals in a process called quorum sensing. These chemical signal molecules are continually made and secreted by individual cells. Once the population reaches a certain number, analogous to an organizational quorum, the chemical signal reaches a specific concentration that the cells can sense. This concentration triggers genetically regulated changes that cause cells to bind tenaciously to the substrate and to each other. Quorum sensing may influence the amount of extracellular matrix produced by the cells. This thick, slimy material consists of polysaccharide polymers called exopolysaccharides and entrapped organic and inorganic materials. The extracellular matrix protects the cells from damage from environmental assaults. Quorum sensing serves the biofilm in many ways, such as by increasing the resistance of biofilms to antibiotics. Quorum sensing in Pseudomonas aeruginosa plays a role in the maturation of the biofilm into complex three-dimensional forms, such as columns and streamers, forming channels through which nutrients flow. Sessile cells in a biofilm chemically talk to each other in order to build microcolonies and keep water channels open. Biofilms tend to form where nutrients are plentiful and may begin to dissolve when nutrients become limiting. Little is known about how a biofilm dissolves, although the process is thought to be triggered by starvation. Pseudomonas aeruginosa produces an enzyme that can strip away the exopolysaccharides. Cells leaving the biofilm revert to their flagellated planktonic forms and can swim to new locations where nutrients are more plentiful. Pseudomonas aeruginosa can be medically devastating. It forms biofilms in susceptible individuals, such as in the lungs of patients with cystic fibrosis.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Danish bin Muhammad Izailan. My metric number is E24A3425. Today I will talk about the case study in the nutrient resource field. The case I going to talk today is the case of the water crisis in the city of Phil in the US. The Phil water crisis was a public health crisis that began in the 2014 after drinking water for the city of Phil was contaminated with a land and Legionella bacteria in April 2014. The impact of water crisis on city of fifth resident were affected with Legionella bacteria will cause them to get Legionella disease when they drink water from the city of fifth, because the water have affected with Legionella bacteria from the biofilm. The bacteria that caused Legionella disease in the city of Fifth was the Legionella bacteria. Legionella bacteria is bacteria that can cause infection to the human. Legionella bacteria live in water areas such as river and the artificial water system, and that's why it can be found in city of Fifth, because they have artificial water system there, and Legionella bacteria grow in the biofilms also.
Okay, Assalamualaikum warahmatullahi wabarakatuh. My name is Alif Syahmi bin Abdul Malik Al-Faisal, and my metric number is E24A3506. So today I can explain about application in the natural resource field. Biofilm is a thin layer of microorganisms such as bacteria or algae found on curtain surface such as where a man-made or non-man-made surface and inside the bodies of living beings. Biofilm are important in the context of natural resource management because they can be utilized for various applications in the field of ecology, industry, and natural resource conservation. Biofilm can be used for the water management and waste treatment. For example, biological filtration. Biofilm are used in water treatment system to reduce organic matter nutrients such as nitrogen or phosphorus and other pollutants. Biofilm can also used for water system restoration. Natural biofilm can help restore water ecosystem by improving water quality through the process of pollutant dehydration. Biofilm can also cause problems in the food and the range industry, medical equipment, and water supply system. Preventing the formation of biofilm is important to ensure cleanliness, including by performing periodic cleaning and sanitation. Routine cleaning of equipment can remove organic waste that become medium for biofilm growth. Monitoring and controlling operation parameter can also prevent the formation of biofilm, such as speeding up the flow. Controlling the flow rate of liquid in the piping system can prevent the adhesion of microorganisms. A proper equipment design is also important in preventing biofilm, such as smooth surface. The use of smooth surface material such as stainless steel can reduce the adhesion of microorganisms. Biofilm are also used in industrial application, including the food and beverage industry, such as fermentation of microorganisms. Biofilm help the fermentation process for the production of products such as yogurt, cheese, and beer. Medical industry, production of antibiotic. Biofilm are used for the production of antibiotic by streptomyces microorganism. Maritime industry, biofilm control on ship it antimicrobial paint to prevent the growth of biofilm and marine organisms on ship hull. In conclusion, understanding biofilm not only help improve the effectiveness of existing technology, but also reduce negative impact on the environment.